Sorry about the focus, folks. I will take care of that from this point forward. What's up, guys? Let's have a lecture on the nervous system. Or nervous tissue, I suppose. But the way this fits together is as follows. What you have to consider is that everything you've ever done, everything, can be broken down into some sort of sensory input, followed by integration in some level of control center, be that the brain or elsewhere, a control center, followed by some sort of what we call a motor output. Sensory input coming in from your sense organs, this would be a stimulus, coming in, being integrated by a control center, followed by some sort of motor output, okay, a response. Here's a classic example. I don't care how fast you're driving, if you're driving down the road and you come around the corner and you see this, that is going to be a sensory input. That sensory input goes to your brain via your optic nerves, and your brain thinks to itself, you know what, I don't feel like being pulled over today. And that is followed by a motor output to remove your foot from the gas pedal. I don't care what the story is, it's going to happen. And you can see it here as well. Here we have a tiger. Here's a guy posing for the best Facebook profile picture of all time. And the tiger gives him a sensory input. The tiger moves. The guy integrates. He doesn't want to die. So he has a motor output to dive out the window. Alright? Sensory inputs, followed by integration in a control center, be that the brain or elsewhere, followed by a motor output. Now, this is a very, very important slide. Alright? A very important slide. And I'm going to run through every bit of the parts and pieces here. And I'm probably going to jump ahead through multiple slides and kind of ruin their concept. I feel like in our current situation, it'd probably be better just to talk about it all here. So I'm just going to do everything here, and then we'll probably have to skip through a bunch of slides as we progress. Alright, here we go. So, your nervous system is broken down into two parts. These are considered the central nervous system and the peripheral nervous system. The central nervous system is contained within bone. This is the brain and spinal cord, okay, protected by your skull, protected by your vertebral column, brain and spinal cord. And then the peripheral nervous system is everything outside of the brain and spinal cord. So here's the brain, here's the spinal cord. This would be the central nervous system. And then all of these peripheral ganglia and nerves, all of that, that's all peripheral nervous system. Very important for us. Now, have we talked about the brain? I honestly forget what all we've talked about. I think we've just done muscle. So let me just outline all this for you. The brain is the main integrating center. The spinal cord tends to carry information to and from the brain. And then we have the peripheral nervous system. Now, you'll see that it's broken down into two parts. Sensory and motor. Sensory carries afferent messages into the brain and spinal cord. Right? The sensory side takes information into the brain and spinal cord, whereas the motor division carries information out from the brain and spinal cord. Sensory in, motor out. Okay, this is very important. I realize how simple that sounds, but people screw it up all the time. Now, these two divisions are further broken down into somatic and visceral. So you'll have a somatic sensory and a visceral sensory, a somatic motor, and a visceral motor division. The somatic sensory division, anything soma means body, anything of the body that we tend to be aware of. So like I can hear, I can see, I can taste. That's all of the body. That's somatic sensory. I'm aware of it. I can feel this table. Okay, I can feel the stretch in my musculature from picking something up. That is all somatic sensory. I'm directly aware of it. Visceral sensory information, it comes from the viscera or the organs typically. Okay, think deep sensory. So I can pick up blood oxygen levels using my kidneys. Am I directly aware of my blood oxygen level using my kidneys? No, I'm not. But that happens. It happens as a sensory mechanism and then conveys that message elsewhere for integration. I'm not directly aware of it. Maybe my body picks up that I am low on water. My osmotic balance is off. I am not directly aware of that consciously per se. That is a visceral sensory bit of information. Another kidney example. So your kidneys will regulate salt levels within your bloodstream. Are you directly aware that your salt levels are high? No, you're not. It's going to happen behind the scenes. You're not directly, consciously aware of it. It is a visceral sensory input. And then there is the motor division. Again, motor is doing work. Somatic motor, soma of the body. The only thing you can control consciously, this is conscious, the only thing you can control voluntarily is skeletal muscle. Okay, skeletal muscle is the only thing controlled by the somatic motor division. You get up, you walk around, you speak, you do the things that you do on a daily basis. That is somatic motor. Visceral motor is outputs to things you are not directly in control of. Like, you don't directly mentally control, you don't decide if your heart beats. You don't decide if you release hormones or not. You don't decide if your intestines are going to be motile. You don't decide where to vasoconstrict and where to vasodilate blood vessels within your body. This is all done behind the scenes. And as you can probably tell, the visceral motor division is vastly more complicated. There's just a lot more happening here. And because it's so complicated compared to somatic motor, because visceral motor is so complicated, we apply a new term set to it. We say that the visceral motor division is your autonomic nervous system. Autonomic. Auto means self, nomos means governing, the self-governing system. This takes care of all homeostatic functions behind the scenes. Is your blood chemistry stable? That's the autonomic nervous system. Okay? How's your temperature regulation going? That's the autonomic nervous system. In fact, this is so complicated that we further break down the autonomic nervous system into the sympathetic and parasympathetic divisions. Now, let me say this first. Both of these work together constantly on a daily basis to keep you alive. The sympathetic division and the parasympathetic division, they are always working with one another to raise your heart rate if you get up to walk around the room, or to drop your heart rate if you sit down and relax, to increase gastric motility or to decrease gastric motility, to increase general metabolic rate or decrease general metabolic rate. These two divisions typically work against one another, that they can work together. They typically work against one another to keep you homeostatically balanced. Now, in extremes, what we call these, the sympathetic division gets called the fight or flight response. So when you think about an emergency situation, like this poor guy here freaking out and running out that window, it's fight or flight, sympathetic division, whereas the parasympathetic division is thought of as being rest and digest. Like, you go out and you eat a big meal and you feel like just collapsing into the chair and sleeping for hours. That is the parasympathetic division diverting blood flow into your intestinal tract to help you digest food. That is rest and digest. Okay? And again, these fight or flight, rest and digest, these are extremes. Normally these work together constantly. Alright, your nervous system has two major subdivisions. Again, as I said, I probably have already talked about all of this on this slide, because I think it's easier just to do everything here. Okay? So let me just click through and we'll see what we get to and what we don't. Alright, central nervous system, brain and spinal cord, and the dorsal body cavity enclosed by the cranium and the vertebral column. That's facts. Peripheral nervous system is made up of the nerves and ganglia. Nerves, do I have an image for nerves? I do, good. So nerves are big bundles of axons. So you know that a neuron, which is a nerve cell, has dendrites, a cell body, and an axon. A nerve is not a microscopic thing. When you hear somebody say, oh, they got a nerve cut, or they have a nerve issue, this is a nerve in a cat's leg. Okay, they are big, they are substantial, all right, and hard too. Like they're tough. It's hard to pull them apart. A nerve, a given nerve, has an appearance like this. It looks almost like muscle. It's got an epineurium surrounding what are called fascicles. Fascicles surrounded by perineurium, and then inside there will be individual axons surrounded by what we call endoneurium. So it looks a lot like a muscle. It really looks a lot like a bone too, with their osteons and lamellae and all that fun stuff. So that is a nerve. Nerves are big bundles of axons. A ganglia is a swelling next to the spinal cord in most cases. Ganglia, ganglia, ganglia. where you find nerve cell bodies of the peripheral nervous system. Okay, so the neurons that go out to your sensory organs, out in your fingertips, for example, they are going to have their nerve cell bodies situated in ganglia right next to the spinal cord. So a ganglion is a knot-like swelling in the nerve where the neuron cell bodies are concentrated. Perfect. That's a nerve. This is a beautiful image from a bodies exhibit I went to in Charlotte. What you see here are all the nerves of the body, these major nerves. These are big, macro. Like you can see the cranial nerves coming down to the diaphragm to innervate the diaphragm here. Just a beautiful thing. Amazing, amazing neural connections there. And again, these are macro. They're big, like the size of a pencil. They're not little microscopic things. Alright, neurons have three major properties. This is very important. They are excitable. That means that they can respond to stimulation, like neurotransmitters. They can respond. They are conductive. They're capable of sending a message from one place to another. And they are secretory. When a message arrives at the end of an axon terminal, it's going to release a neurotransmitter in skeletal muscle. We talked about acetylcholine, if I'm not mistaken. So, excitable, conductive, and secretory. What you're going to find is this is quite similar to muscle. Like muscle is excitable. Muscle is conductive, right? You stimulate one side of a muscle, the stimulus flows to the opposing side. The variation here is that neurons are secretory. They can release neurotransmitters. Whereas muscle is contractile. It's capable of shortening. That's the key characteristic difference between muscle and nervous tissue. Oh, speaking of a neuron. So here's our given neuron. You know that neurons have a nerve cell body. They have dendrites and they have an axon. I'm going to go into a little bit more detail on that now. Alright, neurons are big, special, conductive cells. They're long-lived. They've been alive as long as you've been alive. Some of them since you were in the uterus, okay? These are long-lived cells. They don't, they're non-mitotic. They don't divide and make more of themselves, so they have to live a really, really long problem. Now, why is it an issue that they're non-mitotic? Well, that means if you get nerve damage, is it ever coming back? No, typically not. Especially in the central nervous system, you can't get brain damage and the brain just heals magically. It's not the way this works. There are some little tweaks that can happen, but it's very limited. I will, however, say this. There are mitotic neurons in the hippocampus of the brain. And the reason that there are mitotic neurons in the hippocampus is because this is the area where you store memory. Okay, memories are stored in the hippocampus, so it has to have mitotic neurons there for producing new ones. And I'll say that nerves have an incredibly high metabolic rate. So the first thing that dies when tissue goes anoxic is the neurons. The neurons have to have lots of oxygen to keep up with their metabolic demands. They are very, very, very active. Alright, dendrites, cell body, axon. So dendrites are very short, small little projections. Sometimes they have little branches like this here and even spines that stick up to increase their surface area. Dendrites are for receiving messages. So they are, if you release a neurotransmitter, that will bind to the dendrites, and the dendrites will receive that message and create what's called a graded potential that can then travel through the nerve cell body. Okay, the nerve cell body is called the soma. Okay, soma, body. Nerve cell body is the control center of the neuron. And let's see, what do I want to say here? It basically is the integrative center. It will decide if graded potentials make it all the way through to the axon or not. The axon is going to originate at a structure called the axon hillock. If I were you, I would also know that is called a trigger zone. The trigger zone, or the axon hillock, is very important because basically you stimulate the dendrites, that stimulus flows over the dendrites, gets into the nerve cell body, travels through the nerve cell body, and if it hits the axon hillock, sets off an all or nothing reaction that sends an action potential. Do I have action potential on here someplace? Regardless, it stimulates an action potential that blasts through the axon, just flying through there until it gets to the axon terminals, and that's what stimulates for the release of neurotransmitters. Alright, so graded potential from stimulation of the dendrites flows slowly, graded through the nerve cell body, decides if it's going to continue, gets to the axon hillock, hits the axon hillock, stimulates the axon hillock, which fires an action potential down the length of that axon, hitting to the axon terminals, releasing neurotransmitters. Alright, Let's see, what else do I want to say here? Axon helix, very important. All right, a couple of more comments. So the nerve cell body houses the nucleus, and the nucleus houses the DNA of the neuron. That's a fancy way to say that if there's anything the nerve cell needs, like protein-wise, it has to be made in the nucleus and then carried wherever it has to be. So if I need proteins or whatever way on down here, I got to make it up here and send it all the way down the length. Axons lack the machinery for protein synthesis. So what you have is a two-way street where you can carry big stuff down the length of the axon. This isn't nervous transmissions. This is like big things that have to move, okay? It's referred to as axonal transport. Now, in the realm of axonal transport, there is anterograde motion and retrograde motion. Anterograde motion is movement down the length of the axon towards the axon terminals. Retrograde motion is movement back up the axon from the axon terminals towards the soma. Okay, anterograde and retrograde motion. They have two very special motor proteins called kinesin for anterograde motion and dynein for retrograde motion. And what these will do is you'll make like a vesicle of something that has to move one direction or the other. And one of these transport proteins will bind to the vesicle and literally, I kid you not, walk down microtubule tracts. This is real. Walk down microtubule tracts almost like a myosin molecule can. They look much like myosin. Walk down the microtubule tract almost like it's a railway station carrying that vesicle wherever it needs to go. And this reminds me of that movie Old Yeller. Let's see what time it is. I got to keep monitoring my time so I don't close this video up too soon. It reminds me of the movie Old Yeller. So in this film, a dog gets infected with rabies and they have to isolate the dog for months, and eventually the dog develops rabies and dies. This is a bad infection, viral disease. The reason the dog didn't die immediately is because the actual virus gets into the neurons, into the basal ends here at the axon terminals, gets on a dynein motor protein, and is carried back up the length of the axon of the nerve cell bodies, where it eventually gets into the central nervous system and leads to major central nervous system damage. It's when the animal gets very aggressive and destructive. Yeah, so it's very slow because this process of moving things down the length of an axon is very, 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 very slow. Moving things through the axon is very slow, so these are very slow in process. Now, let's reset our video here. Okay, we have 4 different types of neurons that we're going to discuss here. Multipolar neurons look like this. Bipolar neurons have 2 poles. Bipolar look like that. Unipolar neurons, which appear to have a single projection out of the nucleus that goes 2 different ways. And then anaxonic neurons. Anaxonic means no axon. Okay, so let's talk about what this does. Multipolar neurons, oh man, very common. These are like 99% of the neurons in your body. This is the type of neuron you find in your brain and spinal cord. Like, think about this, right? So you've got sensory neurons and you've got motor neurons. Sensory information comes in via sensory neurons. It's integrated by the brain, and then motor neurons go out. Where would you find more nerves? In the brain and spinal cord, because it's in the brain that you're figuring out what the sensory information means. It's in the brain where you figure out what motor output needs to happen. The multipolar neurons here in the brain and spinal cord are way more numerous. 99% of neurons in the body are multipolar neurons. So these are very, very, very prevalent. And this is what your given neuron, your general neuron looks like from our perspective. Bipolar neurons are much more rare. Bipolar neurons, again, will have dendrites. Or let me rephrase, a dendrite and an axon is the way we think about this. A single dendrite and a single axon as a bipolar neuron. These are typically going to be found in your special senses, like your eyes have bipolar neurons that connect one spot directly to another spot. Your nose has bipolar neurons that connect your olfactory epithelia directly to your brain. Basically, when it needs to go from one place directly to another place, that's bipolar neurons. Okay, that's what you're going to use. Retina, inner ear, you know, what have you. They're special senses. Then there are unipolar neurons. Unipolar neurons aren't truly unipolar. What they are is during the development process, the axon and the dendrite fuse at one tip, so you end up with this one little spot here connected to the soma, the nerve cell body. But in reality, these are dendrites coming in, and attaching, and then that's an axon coming out and attaching down here. So it's really more like a bipolar, but we call them unipolar because they have one projection coming off the nucleus. All right, so what are these? These are the neuron of the peripheral nervous system. So when we talk about these big old ganglia and things that we were doing here, these ganglia and all these nerves coming out and all the axons coming through here, those are all going to be unipolar neurons. They're all unipolar neurons. And then last but not least, anaxonic neurons. So they lack axons. So if they lack axons, what can they do? The idea is that these are found in the visual process, where when your rods and cones or bipolar neurons are releasing neurotransmitters in your eyes for you to be able to see, anaxonic neurons only have dendrites. So what they do is they take up excess neurotransmitter to fine-tune the images that you see, to fine-tune the way in which neurotransmitters are being released. So those are anaxonic neurons primarily for visual processing. Alright, functional classes. Sensory neurons, interneurons, and motor neurons. We've kind of already played this game. Sensory neurons carry afferent messages to the brain and spinal cord. Interneurons are for integration. They make up the vast majority of neurons found here. And then motor neurons carry messages out to do some sort of work. They carry what we call efferent messages. If I were you, I would know my terms afferent and efferent and realize that most neurons are integrative. Making decisions is much harder than perceiving or sending messages. So the vast majority of neurons are considered interneurons. Alright, let's talk about the neuroglial cells. So neuroglia means nerve glue. Neuroglial cells are the helpers. Okay, you got to think about neuroglia as being the entourage of the neuron. Okay, the entourage of that neuron. If the neuron releases waste, the neuroglia cleans it up. If the neuron gets infected by something, the neuroglia clean it up. If the neurons need cerebrospinal fluid, the neuroglia makes cerebrospinal fluid. It's the neuroglia that keep the central nervous system and the peripheral nervous system working. And these neuroglia are specifically of the central nervous system. I will talk about 4 main types here. These are the astrocytes, microglial cells, ependymal cells, and oligodendrocytes. Those are all good terms to say. Oligodendrocytes. Now, you have seen neuroglia before. When you looked at photomicrographs like this, you could see the little dots. Like this is a good neuron. These little dots, these are neuroglia. Okay, those are neuroglia. That's a neuron. These are either axons or dendrites. Okay? So let's talk about it. Astrocytes are the catch-all. Okay? They do lots and lots of things. They will move the neuron around if the neuron needs to move. They will help get the neuron where it needs to be to synapse with specific cells. If the axon releases too much neurotransmitter, the astrocytes clean it up. If the axon doesn't release enough neurotransmitter, the astrocytes can release some. Astrocytes are the helpers, the general helpers. Alright, microglial cells take the place of white blood cells in your brain. Your blood does not allow white blood cells into your brain. Okay, you don't do it. There's what's called a blood-brain barrier. You don't let white blood cells in the brain. So how do you protect the brain? You use microglial cells, special supportive neuroglia that act as protective cells. Okay, those are microglial. Ependymal cells, mainly associated with a structure called choroid plexus. Ependymal cells are associated with choroid plexus. They are ciliated, and what they do is they make and move cerebrospinal fluid. Okay, they make and move cerebrospinal fluid, do ependymal cells at specialized structures called the choroid plexus. And then last but not least, there are oligodendrocytes. Oligodendrocytes make a special substance called myelin. They make what's called the myelin sheath. Okay, the myelin sheath. Basically, myelin, as you can see here, this is a oligodendrocyte wrapped around some axons here, nerve fibers. The oligodendrocytes with myelin insulate the neurons, insulate their axons so they can transmit messages faster. An insulated axon works better, and that's what myelin does. All right, it insulates the axons. And you can actually see it in the brain. So you've probably heard somebody before say gray matter and white matter. So the gray matter in the brain, which is kind of this darker color on the outside mostly, this little bit in the middle, the darker color, this gray matter, is where you find the nerve cell bodies, and the white matter is going to be oligodendrocytes, myelin sheaths wrapped around axons. So these are all axons wrapped in myelin. And it kind of makes sense that it's light in color, white matter. It's based off of a lipid. Myelin is based off of a lipid. So if you go and you look at, like, geez, I don't know, ribeye steaks at Publix or something like that, and you look at the fat is very light in color. It's almost white. That's the same reason this is very light within the brain, because myelin is based off of a lipid. Alright, then there are, let's see, okay, so this is the peripheral nervous system. In the peripheral nervous system, the two cells that I'm concerned with are called Schwann cells and satellite cells. Satellite cells simply insulate the nerve cell bodies in these anaxonic neurons. What am I talking about? Unipolar neurons. And then we have Schwann cells. Schwann cells are the important part. Let me say it again. Schwann cells are the important bit of this. The Schwann cells insulate axons. And what you have in the peripheral nervous system is all these Schwann cells wrapped around the axon, and they have little gaps in between them. Gap, little gaps between the Schwann cells. And the gaps are called the nodes of Ranvier. Let me say that again. Schwann cells are found in the peripheral nervous system. They cover and insulate the axons in the peripheral nervous system. Sometimes they don't. And where they are, there will be little gaps between cells called the nodes of Ranvier. The nodes of Ranvier. This is very important to how this all fits together. Okay. So, conduction speed of a nerve fiber. There are two ways to make a nerve fiber conduct messages faster. One is to increase the diameter of the fiber. The bigger the fiber is, man, hey, the faster it's going to conduct messages. Big fibers send messages quicker than do small fibers. That's just how a nerve works. And then the best way to speed up the transmission speed of a nerve fiber is by putting myelin in place. If you've got myelin and it's bigger in diameter, boy, it just really jets. Okay, it sends messages incredibly fast. So where would you have some and where would you not have some? The idea is that you just don't need myelin everywhere. So think about your nose, right? The olfactory epithelium is right at the very roof of your nose, and your olfactory bulb is right above that, like just about a ½ inch. And then the area where you interpret smell is right next to that. You don't need myelin there. That transmission speed is going to be really high anyway because it's such a short distance. But your touch receptors in your feet, the stretch receptors in your leg muscles that keep you upright, make sure you don't fall down, they need myelin. Okay, they need myelin so they can conduct messages very fast. Like the neurons that regulate your intestines, they don't need myelin. They don't need to expend the energy for myelin, for Schwann cell support. They don't need it. These are going to be, you don't have to have instantaneous transmission speed. But your skeletal muscles that help you with your posture, they're going to have myelin so they can have an incredibly fast transmission speed. That's just how it works. If you want to be able to stand up straight and walk around, and if you trip, catch yourself, you need myelin to make sure those nerve transmissions are as fast as possible. Alright, signal conduction. So there are two forms of conduction here. There are unmyelinated neurons, and then there are myelinated neurons. In a myelinated neuron, what you have is what's called continuous conduction. Do I even have that written here? I don't. Well, let me tell you, it's called continuous conduction. I can't believe I don't have that written down. Anyways, it's referred to as continuous conduction because what will happen is you'll depolarize an area past the axon hillock, and when there's no myelin, the stimulus, the change in ionic charges that's going to happen here, called a action potential, flows down the length of that axon continuously. So basically, the stimulus is going to stimulate this area, then this area, this, flowing down the length of that axon from one end to the next continuously, not skipping anything in between. When myelin is in play, what we call the conduction is saltatory conduction, or saltation, saltatory conduction. Saltation means to leap or to jump. So saltatory conduction, what you're going to do is, instead of having a continuous flow down the length of the axon to convey that message, is you're going to have the stimulus jump or leap from node of Ranvier to node of Ranvier to node of Ranvier to node of Ranvier. So what you're going to do is, instead of going all the way down the length, it's going to go boop, 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 done. it will skip all the area around the Schwann cells. It will jump from node to node, skipping everything in between, making the transmission speed really fast. All right, really, really fast. Again, you're skipping all the area underneath the Schwann cells, jumping from node to node. This is called, and you're going to see this again, saltatory conduction. Saltatory conduction, jumping from node of Ranvier to node of Ranvier. And let's have a little bit of a conversation on regeneration of nerve fibers. The main thing I want to get across to you here is... Hang on, let me check my time. Yeah. All right, the main thing I want to get across to you here is that for you to heal... okay, if you damage a nerve cell body, it's never coming back. Period, the end. If you destroy that nerve cell body, it's dead. The nerve's dead, it's over. If you damage an axon in the central nervous system, that nerve's done. It's never going to work again. It's over with. The central nervous system does not heal. That's why if you get a nerve spinal cord injury, it's over with. But in the periphery, in your hands and your arms and your legs, okay, if you were to damage a nerve in your arm here, like an axon, if you hit the axon here in the arm that controls that finger, you can heal that. That can come back. It's a doable process, especially if it's a myelinated neuron. Now let me show you how this works. If you were to cut a neuron, so you slice it through there, you destroy that bit of the axon, the nerve cell body's intact, so we're good. What will happen is everything downstream of the area of damage, everything downstream dies. And what you'll do is, the nerve cell body is going to get real big, and it's going to start cranking out protein, and it's going to release all these little feelers off the end of the axon that's left, and they're going to move all through your tissues looking for their point of synapse. And when they find that point of synapse, they set up shop, and all the other peripherals fall off, or they degenerate, you might say, and the nerve will be healed. It'll be functional again. So what's the importance of Schwann cells? Well, if the axon dies, the Schwann cells stay alive. And they form what's called a regeneration tube. A regeneration tube. This regeneration tube, the axon will grow into it and follow it all the way back straight to the initial point of synapse. All right, straight to the initial point of synapse. So by having Schwann cells in place, we can vastly increase the speed of healing in this process. Now, this takes a long time, weeks, months, for this to occur. Really long axons, like my brother, it took him like 2 months. These grow very slow, a millimeter and a half per day. And when you're talking about, this much axon you got to regrow, that takes a long time to do. So this is a slow process, but in the peripheral nervous system, especially when Schwann cells are present, it's possible. It's possible. All right, and that takes us on to axon, or rather to action potentials and other interesting stuff. So I'm going to, let's say we pause it here. So pause it there, and then pick it up on the next one. Like take a little break and pick it up from here in a bit. Back we are. Alright, so action potentials and other interesting stuff. Alright, so electrical potentials and currents. The idea is that what we're going to be dealing with when talking about nervous transmission is the movement of charged ions. These are sodium and potassium. Everything's graded potentials, action potentials, it's all based off of the opening and closing of gated ion channels and the exchange of sodium and potassium, leading to what we call a depolarization wave and a repolarization wave. So it's all about storing electrical charges using what we call a sodium-potassium pump to store sodium and potassium where they're supposed to be, and then when the nerve depolarizes, those ions exchange places through gated ion channels, not unlike how skeletal muscle works. So if you know how skeletal muscle works, you kind of know how this works. Alright, living cells are polarized. If you could measure the charge on a neuron at rest, its charge would be about negative 70 millivolts. Now, there's a reason for this. Inside, hang on, I might be jumping ahead. I'm probably jumping ahead. Yeah, I'm definitely jumping ahead. Okay, let me just make this very simple. You have a charge on neurons based off of the presence of sodium and potassium in opposition to one another of about negative 70 millivolts on a given neuron. Okay, that's at rest, negative 70. Now the main reason for this is because inside of a neuron, all the big proteins, all these big organic acids and things, all the whatever, it's all quite negatively charged. All the stuff in there is negatively charged. So what will happen is potassium and sodium are capable of exchanging places and changing the charge. Okay, that's really all I want to say here. Sodium and potassium have the ability to exchange places and augment the charge on that neuron. It can go from -70 all the way up into the positive end of the spectrum. Now, the way that we keep sodium and potassium where they're supposed to be, again, is using our sodium-potassium pumps. That is going to make up about 70% of the energy requirements of the nervous system, is all in sodium and potassium being pumped into their concentrations on either side of the cell. Of the axon membrane is what we're really talking about here. Okay, now then, on to the point. So let's talk about graded potentials and action potentials. So graded potentials are local disturbances. They move over short distances on the dendrites. Action potentials are long-term changes in the membrane potential acting over long distances, dealing with the axons primarily. Yeah, perfect. Alright, so graded potentials. What do I want to say? We sort of already have done this. I'm just going to run you through it very simplistically. The idea is when you stimulate the dendrites of a neuron, it's going to stimulate for a graded potential, primarily based off of sodium exchange. Sodium is opened up, and the sodium channels open up, and sodium rushes into the cell. It's kind of complicated, but the idea is that sodium rushes in, and that sets off a chain of reactions that flow through the dendrites, going into the soma, and hopefully striking the trigger zone, signaling for an action potential to then go down the length of that axon. The reason we call these graded potentials is because they're decremental. Let me say that again. They're graded potentials because they're decremental. They will get weaker over time. You can have a really strong, these are graded, a really strong stimulus on that dendrite, and that stimulus is going to get weaker and weaker and weaker and weaker and weaker, and then eventually hit that trigger zone and stimulate it to go the whole length of the axon as an action potential. Or alternatively, you could have a weak graded potential signal from the same place. And a weak graded potential is going to get weaker and weaker and weaker and die out prior to reaching the trigger zone. So key point, not every graded potential results in an action potential. Not every graded potential results in an action potential. The idea is if you can stimulate that dendrite strong enough to get the stimulus all the way down to the trigger zone, then the trigger zone will trigger an action potential. Action potentials that look like this, by the way. So action potentials follow what we refer to as an all or nothing law. The action potential is either going to start and flow down the length of that axon, or it's not going to. It's dominoes. They either go or they don't go. That's just how this works. They are non-decremental. They do not weaken over time. It's all or nothing. You get the spike. You can see the spike going down the length here. This is continuous conduction. It's like if I had a big long rope and I could put a wave in this end, and you can see the wave shooting down the length. That's an action potential. It's going to shoot down the length and exchange in charge, going from -70 all the way up to the positive side of the spectrum. All the way down the length. Boom, 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 boom. This is the opening of gated ion channels. Ions, 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 ions. All the way down the length of this. That is an action potential. Triggered by the axon hillock or the trigger zone. Right. That's exactly what I'm saying here. Gold vein... Let's see, gated ion channels, voltage regulated, the soma, so you have a stimulus on the dendrites, which is a graded potential. It's going to flow down the length of this, eventually hitting the trigger zone. The trigger zone is special because it's just packed with gated ion channels, and when they crack open, they stimulate the action potential that goes down the length. So the trigger zone is important because it's the end of the soma, and when you stimulate that trigger zone, you're going to stimulate for an action potential that travels the whole length of the axon. Now, let's talk about what the action potential looks like. If I could come in here with a very special probe and insert it into that axon, like insert it into a nerve going to a muscle or something like that, when you send a stimulus down the length of that nerve, what you see is this spike I was describing earlier. A spike from -70 millivolts up to the positive end of the spectrum, and then dropping down below the original line, and then coming back to a resting state. We call this a wave of depolarization, followed by a wave of repolarization, followed by a brief hyperpolarization period, followed by a return to a resting state as a result of sodium-potassium pumps pumping everything back to where they're supposed to be. So let's describe it. The wave of depolarization, this is going to be caused by sodium channels. Sodium channels pop open in unison, and you have this huge spike in charge because all these positively charged sodium ions come flooding into that cell, and as they come flooding into the axon at that spot, it's going to spike the charge up to the positive end of the spectrum. Now, you crack open your potassium channels, and potassium leaves the axon. Potassium's positively charged, so as the potassium ions leave, the charge goes back down, all the way to the negative end, because potassium channels are very slow to react, so they slowly close, and basically they lead to a hyperpolarization or a refractory period, and then sodium-potassium pump everything back where it's supposed to be, and we get back to our resting state. Now, this refractory period is very important. What this means is that your body is capable of telling the difference between two very quick succession action potentials by using a refractory period. You can't just stimulate an axon and leave it stimulated. The stimuli have to go bam, 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 bam in waves, because the refractory period basically tells the body, okay, that's the end of one, prepare for the next one. So you can get spike, refractory, spike, refractory, spike, refractory. The refractory period is very important for separating out these action potentials. Yeah, a wave of depolarization followed by a wave of repolarization followed by a return to a resting state using sodium-potassium pumps. Alright, now then, a nerve signal can go no further once it reaches the end of the axon, that axon terminal. When you have a stimulus go down the length of the axon, hit the axon terminal, what it does is it cracks open synaptic vesicles, releases neurotransmitters into the synaptic cleft, which then bind to what we call a postsynaptic neuron, stimulating that postsynaptic neuron. It's very important to point out here that this is an electrical signal, followed by a chemical signal using neurotransmitters, followed by another electrical signal moving down the length. So when we say like a sensory neuron stimulates an interneuron, and that interneuron stimulates a motor neuron, what we're having there is an electrical signal followed by a chemical signal, followed by an electrical signal, followed by a chemical signal. These nerves do not directly touch one another. They have to have neurotransmitters that are capable of being exchanged for that synapse, for them to synapse. And some nerves can have crazy numbers of synapses, man, enormous numbers. So like spinal motor neurons can have 10,000 synapses. So one nerve connects to 10,000 others. In your cerebellum, which is a crazy structure to begin with, there can be 100,000 synapses per neuron. That's a huge number of nerves connecting. So it's not just one-to-one. It's one to lots. It's like a big old Christmas tree of connectivity, man. Yeah, that's perfect. Okay, there are a lot of different neurotransmitters, and I think I'm going to talk about a few. Yeah, okay, so 4 major suites of neurotransmitters: the acetylcholine neurotransmitter, amino acid-based neurotransmitters, neuropeptides, and bioamines. I'm going to keep this very simple. Acetylcholine is acetylcholine is acetylcholine. It gets a group, a category all to itself. This is going to be stimulatory for skeletal muscles. We've talked about it before. Amino acid neurotransmitters include things like glutamate, aspartate. These tend to be like GABA is quite famous, gamma-aminobutyric acid, as an inhibitor. This is used in certain drugs for like inhibiting muscular activity, like cramping, for example. So we will use these neurotransmitters as an inhibition method to decrease responses of certain tissues in the body. Now, the next two are way more interesting from my perspective, and that is the neuropeptides and the bioamines. The reason these are fascinating, let me check my time. The reason these are fascinating is because they are neuromodulating. Let me say that again. Both of these, neuropeptides and bioamines, are neuromodulators. What that means is they bring about a whole body-wide augmentation. Your entire physiology changes when these are in play. So what are they? Well, like endorphins. Okay, I will never forget when my wife had my second child. It was like the easiest delivery on the planet. Like if this was child delivery, like no one would complain about it. She goes into the hospital, sits down. 2 hours later, she tells the doctor she wants to push. She's totally fine. She pushes for about 35 or 40 seconds, and then out comes a baby, and everyone is completely fine and happy. They drop the baby on my wife's chest, and she's like, I just love him so much. I want more, and you're all wonderful people. She was high as a kite on endorphins. Alright? She felt no pain because of endorphins. Pregnancy and delivery is tough, man. That's a for real thing. But she was totally cool during the whole process because of endorphins. Alright? These have an effect on the body of calming you down and decreasing the way you feel pain, just making you feel better, feel good. Okay, you feel real good. Opiate-based drugs mimic the effect of endorphins. Okay, they mimic that effect. Even like comfort food is the way your book would describe it. So comfort foods elicit an endorphin-like response. So you go in grandma's house and you smell that cooking and you just feel better. Like everything's just better. That's a release of endorphins. That's a classic release of endorphins. Alright, those are neuropeptides and neuromodulating, causing a whole body-wide effect. Same thing with bioamines. Bioamines are things like epinephrine and norepinephrine. Think about like adrenaline rushes. That's a bioamine. Dopamine is very important for this. This is one of the catecholamine neurotransmitters. And histamine and serotonin are involved in this as well. But I want to talk about dopamine. Dopamine can cause all kinds of problems with the body, man. So if you've never seen the movie A Beautiful Mind, I invite you to go and watch it. The guy has got some problems that are based on dopamine. He doesn't have enough, I think. He's not making enough dopamine. So the side effect of this is he has very powerful hallucinations. No, backwards. No, correct. I'm totally messing this up. I think he's getting too much dopamine, that's correct. And what that means is he's having hallucinations that seem real, things that seem real. The reason this kind of confused me is because acid, LSD, LSA even, and ergot fungus, these will mimic the effects of too much dopamine in the brain and cause people to see things that aren't real. And we're back. Slowly getting the kinks worked out of this thing, man. I tell you. I think we were done with this. We were talking about these neuromodulating bioamines and neuropeptides. I'm just going to jump over to here. Alright. A few party comments. The type of receptor is just as important as the type of neurotransmitter. Okay, the type of receptor is just as important. It's kind of like your group of friends. You say the exact same thing to all of them, and they all perceive it in a slightly different way. Well, I'm here to tell you that there's 14 different types of receptors just for serotonin. Alright? These neurotransmitters, they can have inhibitory receptors, they can have excitatory receptors. So you can expose some types of muscle to acetylcholine, they are stimulated. You expose other types of muscle to acetylcholine, they are inhibited. So there is a lot of variation that can occur here. So the physiology of the cell can certainly be altered by the type of receptor available. The receptor governs the effect, not the neurotransmitter. The neurotransmitter tells the receptor, hey, it's time, but the receptor determines inhibitory or excitatory or a grade within all of that. Alright? Yeah, which is what I'm saying here. So excitatory and inhibitory just kind of depends on where you are and what you're doing. Right. And we'll talk more about all this in another lecture. Alright, parting comments. Neural processing. Two forms. There are serial processing and there is what we call parallel processing. Now, serial processing, serial means direct, basically. One thing leads to another thing, leads to another thing, leads to another thing. They are in serial order. What this basically is indicating is that in a serially processed event, the way your brain works in a serial processed event, all of you, everyone listening, would have the exact same response to a given stimulus. So if your blood sugar levels drop, you'll take steps to bring your blood sugar levels back up. That is serial. We all do it the same way. If you touch something very hot, you'll pull your hand away. That is serially processed. It's a reflex. Okay? One thing to the next thing to the next thing to the next thing. The integration is very simple. A receptor sends the information. It is integrated and an effect is sent out, the same for all of us. And then there's parallel processing. Parallel processing is seeing the forest, not just the trees. We all have multiple unique pathways. Sometimes my wife will look at me and say, "What are you thinking about?" She can tell I'm on something, and I'll say, "Oh well, I got to take you down this roller coaster to get you where we're going on this." So like I think about one concept, then another, and then another, then another, then another, and eventually I wind up at some weird place, and I have to explain to her how I got there. The idea is that all of us have different life histories, ergo we all interpret stimulus at a different format. We all see things in a different way. I'll give you an example. And bam. Really think about this image set. What do you think when you see this? And I'll tell you exactly what I think when I see this, and I think about comfort food, man. I think about when I was young and my brother first got his driver's license and he'd drive us home from school and we'd go by McDonald's. I think about getting a number 2. That was 2 cheeseburgers with fries and a drink. We'd share the drink and the fries and we'd each get a cheeseburger. That's what I think about. I think about my brother. Like, it takes me back to being 14 years old, all right? For you, this elicits a completely different response because you don't have the same life history as I have. It's parallel processing. It varies from person to person how we think about the world around us. And I think that's it. Yeah, that's it.